Hello everyone. In this video I will be explaining the experiments conducted by Nirenburg, Matthaei, and Letter and how they determine which mRNA sequences code for which amino acid. First, let's start with some definitions. A codon is a sequence of 3 nucleotides in mRNA which together code for an amino acid. Amino acids are building blocks of proteins, and there are 20 of them occurring in the world. Translation is the process of converting mRNA to amino acids or proteins, and transfer RNAs are a type of RNA that physically brings the amino acid to the ribosome where translation is taking place. The first experiment was done by Marshall Nirenburg and Heinrich Matthaei in 1961. First, they radioactively labeled one of the 20 amino acids and put each of them in a mixture with the strand of repeating uracil nucleotides together and incubated them so that translation could take place. The resulting solution was filtered, and the filter was then checked for radioactivity. They found that only the phenylalanine amino acid had the radioactive label on it, signifying that its codon was UUU. Next, they repeated the experiment with strands of C's, A's, and G's, and discovered that C's code for proline, A's code for lysine, and the G's did not work. 3 years later, in 1964, Nirenburg partnered with Philip Letter to determine the codons for the rest of the 20 amino acids. They used multiple short sequences of different known mRNA codons and added them to a mixture with ribosomes and tRNAs, along with one radioactive amino acid per solution. The resulting solutions were filtered like before, but this time the unbound tRNAs were allowed to flow through. The filter was checked for radioactivity each time. If radioactivity was detected, that means that the tRNA bound to the mRNA codon, identifying which codon was for which specific amino acid. Due to their experiment, they correctly identified 61 codons and made scientific history. Marshall Nirenburg actually won the Nobel Prize in Physiology and Medicine in 1968 for his accomplishments.